Assalamu alaikum warahmatullahi ta'ala wabarakatuh. Today in this sixth lecture of lipid metabolism, we are going to discuss cholesterol, particularly its synthesis, transport, and excretion. Our objectives are to list the sources of cholesterol, both in the plasma and tissues, to identify the main roles of cholesterol in the body, to locate cholesterol synthesis at tissue and cellular levels, to discuss the steps and regulation of cholesterol synthesis, to discuss transport and excretion of cholesterol, to identify the role of cholesterol in bile acid synthesis, and to discuss biomedically relevant disorders and drugs. This is cholesterol as you know it. It is a complex structure, and we are going to learn today how the body manages to synthesize it. Cholesterol in our body is either derived from the diet that we have taken it within our food, or we have synthesized it ourselves, so it is synthesized de novo. Cholesterol is present in tissues and plasma lipoproteins either as free cholesterol. When we say free cholesterol, we mean that it is unesterified, and this accounts for around the third of the total concentration, that is 30%. The rest, the 70%, is present as cholesterol esters, meaning that the cholesterol is conjugated with or linked to a long-chain fatty acid. Plasma concentration of cholesterol is 150 to 150 milligram per 100 ml or per deciliter. More than 200 is considered hypercholesteremia, which has serious risk we are going to discuss later on. Main roles of cholesterol include a structural role, for example, as part of a plasma membrane, as part of lipoproteins, or as part of the myelinated structures around neurons. Cholesterol also is a precursor of bile acids, steroids, hormones, and vitamin D. Today we are going to discuss how the body uses cholesterol as a precursor for synthesis of bile acids. Cholesterol also protects the gallbladder membrane from irritation and the harmful effects of bile acids. Cholesterol can be virtually synthesized by any cell in the body. However, at tissue level, the main synthesis sites are liver, intestine, adrenal cortex, and reproductive organs. And at the cellular level, the synthesis occurs in the cytosol and in the plasmic reticulum. So collectively, you can say that cholesterol synthesis occurs in the extramitochondrial compartment. Cholesterol synthesis proceeds through 5 steps: formation of mevalonate, isoprenoid urics, squalene, non-sterol, and finally cholesterol. The first step, formation of mevalonate, occurs by condensation of 3 acetyl-CoA molecules, either by condensation of 2 acetyl-CoAs directly to form the acetoacetyl-CoA, or acetoacetyl-CoA. This for a carbon atom compound can be derived from ketone body synthesis, from ketogenesis. So this acetoacetyl-CoA then condenses the third molecule of acetyl-CoA, catalyzed by the HMG-CoA synthase to form HMG-CoA. And this HMG-CoA is present in the cytosol, in contrast to the HMG-CoA that is part of the ketogenesis in the mitochondrial compartment. The third step is for formation of mevalonate through a two-step reaction catalyzed by HMG-CoA reductase. This step or this reaction is the limiting step in the whole process of cholesterol. This is HMG-CoA reductase can be inhibited by mevalonate, which is the medial product, can be inhibited by cholesterol, which is the end product of the whole synthetic pathway, as well as bile acids. It is also a major action for lipid-lowering agents called statins, particularly the examples include simvastatin. The second step is isoprenoid unit formation by a series of phosphorylation reactions in which ATP is used as a source of phosphate. So mevalonate will be phosphorylated to mevalonate 5 phosphate, which is again phosphorylated to mevalonate 5 diphosphate, which is phosphorylated to mevalonate 3 phospho 5 diphosphate. And the final step is a decarboxylation reaction whereby isopentenyl diphosphate is formed. Isopentenyl diphosphate is considered an active isoprenoid. The third step is squalene formation, also proceeds through several reactions where dimethyl diphosphate condenses with isopentenyl diphosphate to form geranyl diphosphate, which condenses with another molecule of isopentenyl diphosphate to form farnesyl diphosphate, and then farnesyl diphosphate forms squalene. Squalene, as you can see, we are going coming closer to the structure of cholesterol. However, this structure is open and the cholesterol has closed rings. So we have further steps to go through. The fourth step is non-sterol formation. Squalene is converted by squalene epoxidase to squalene epoxide, which is then cyclized by oxidosqualene lanosterol cyclase to lanosterol. Now we are coming even closer to the final structure of cholesterol. The fifth step, which is actual cholesterol formation, occurs in the endoplasmic reticulum, where lanosterol is converted to cholesterol via several steps. Regulation of cholesterol synthesis is via regulating HMG-CoA reductase. The activity of this enzyme is low during fasting, and it is inhibited by mevalonate, as we said, cholesterol and LDL. It also exhibits diurnal variation, so its activity is different between evening and morning. It has a cyclic rhythm, and this affects a timing of the best time for administration of statins. Insulin has a dominant role compared with glucagon regarding regulation of HMG-CoA reductase. Particularly, administration of insulin and thyroid hormones increase activity of HMG-CoA reductase, while the activity of HMG-CoA reductase is decreased by the antagonistic hormones glucagon and corticosteroids. HMG-CoA reductase has two forms, active and inactive. The active form is dephosphorylated, while the inactive form is phosphorylated, and as we said, it is inhibited by the group of drugs statins. At a cellular level, there is what we call cholesterol balance between synthesis and production, between factors that increases it and factors that decrease it. So cholesterol is increased due to uptake of cholesterol containing lipoproteins. So cholesterol here is taken within the lipoprotein. The balance can be increased due to uptake of free cholesterol from cholesterol-rich lipoproteins. Balance of cholesterol can also be increased if there is active cholesterol synthesis, apparently. And the fourth factor is hydrolysis of cholesterol esters by a hydrolase. So a hydrolase will release free cholesterol from the attached or the linked fatty acid to have a free cholesterol, and this will increase free cholesterol level. Cholesterol concentration or level is decreased due to efflux of cholesterol from cholesterol low lipoproteins by L-lysine cholesterol acyl-CoA transferase. This transferase will transfer cholesterol to load it into low lipoproteins, low cholesterol concentration lipoproteins. Esterification of cholesterol will decrease, of course, the free cholesterol level because cholesterol will be converted to cholesterol ester. Utilization of cholesterol by synthetic pathways, steroidogenesis, or for any process that uses cholesterol as a precursor, of course, will decrease the level of cholesterol. How is cholesterol transport in the blood? cholesterol is transported through lipoproteins, particularly the low lipoproteins of low cholesterol potential, like HDL3, and it's also transported by high cholesterol content lipoproteins like LDL. Unesterified cholesterol transfer readily between cell membranes, lipoproteins, and red blood cells. The problem is with the esterified cholesterol. Lysine cholesterol ethyl transferase forms most of the plasma cholesterol esters. Cholesterol ester transfer protein facilitates transfer of cholesterol esters between lipoproteins. How is cholesterol excreted? In bile, particularly as cholesterol or as bile acid for salts. 1 gram of cholesterol is excreted daily. Copranolol is the principal sterol and traces is formed from cholesterol by intestinal bacteria. Most of the excreted bile salts are reabsorbed back into the portal circulation in what we call the enterohepatic circulation. So bile acids are formed from cholesterol and stored in the gallbladder. When a meal enters the body will release some of these bile acids into the small intestine to help in digestion and absorption of lipids. And as the meal goes through the intestine, the small intestine, there is reabsorption of bile acids back into the blood and back into the liver. This is what we call enterohepatic circulation, to be reused again and excreted again in the bile. The fraction of bile acids which are not reabsorbed back into the circulation will be lost in feces. Now we will talk about bile acid synthesis. Cholesterol, as we said, is the precursor for primary bile acids. Primary bile acids are two. They are cholic acid and chenodeoxycholic acid. The enzyme 7-alpha hydroxylase catalyzes the rate-limiting step in bile acid formations, particularly primary bile acids formation. The pathway, of course, divides between the formation of the two primary bile acids. Primary bile acids enter bile as glycine or taurine conjugates. So they are first synthesized as cholic acid and chenodeoxycholic acid. Then each of them can be either linked or conjugated to glycine or taurine to form a conjugate that can now enter the bile acid in a solid form. A portion of, we said, bile acids will be excreted in the intestine to help in absorption of lipids. Part of the bile acid, primary bile acids that are secreted into the intestine can undergo further changes by the action of bacteria in the intestine. The two changes include deconjugation and 7-alpha dehydroxylation. The products now are called secondary bile acids. So secondary bile acids are formed from primary bile acids in the intestine by intestinal bacteria, which carry deconjugation and 7-alpha dehydroxylation processes. What are the secondary bile acids? They are deoxycholic acid, which is formed from cholic acid, and lithocholic acid, which is formed from chenodeoxycholic acid. Bile acid synthesis is regulated at the level of the enzyme 7-alpha hydroxylase as well as the HMG CoA reductase step of cholesterol synthesis. So, cholesterol, by the action of 7-alpha hydroxylase with at hydroxyl group here at position 7 to form 7-alpha hydroxycholesterol. Here we have bifurcation or division of the pathway leading to formation of the two primary bile acids. CoA can be conjugated either to taurine to form taurocholic acid or can be conjugated to glycine to form glycocholic acid. Both of them are primary bile acids. Chenodeoxycholic acid also can either be conjugated to taurine or glycine to form tauro and glycochenodeoxycholic acid, primary bile acids. Bacteria, as we said, act on the primary bile acid to form secondary bile acids. So, taurocolic acid and glycolic acid by deconjugation and 7-alpha dehydroxylation will form deoxycholic acid, which is the secondary bile acid from choline OA. While deconjugation and 7-alpha dehydroxylation of phenodeoxycholic acid conjugates, TOR and glycoconjugates, will form lithocholic acid. So lithocholic acid is the secondary bile acid from phenodeoxycholic acid. We have certain disorders or medical disorders that are related to cholesterol. Cholesterol in serum is correlated with the incidence of atherosclerosis and coronary artery disease. It is whenever the level of cholesterol is higher, or the higher the level of cholesterol, the higher the incidence of both atherosclerosis and coronary heart disease. There is an inverse relationship between HDL concentration and coronary heart disease. That is why HDL is called the pure cholesterol, while LDL, because it is level is directly related to incidence of atherosclerosis, ischemic heart disease, LDL is called the bad cholesterol. A group of conditions called hypercholesterolemias, apparently related to high level of cholesterol in the blood, can be either primary inherited conditions such as familial hypercholesterolemias, or could be secondary due to other causes such as diabetes mellitus or nephrotic syndrome. And this picture here shows a section of a blood vessel in which there is a plaque formation. Atherosclerotic plaque. Normally, the both of the sides or the inner wall of the artery will be smooth like this, but when there is high level of cholesterol, this cholesterol will deposit on the arterial, for example, wall, and this will lead to abnormalities of the vascular endothelium and inflammation, followed by deposition of monocytes, which mature into macrophages, which will attract more cells with, on top of which there is deposition also of platelets. So a plaque will be like this. It can get bigger, it can lead to stenosis and narrowing of the lumen, or it can detach, because after some time the structure will be loose. Detachment of this plaque is very serious because it can travel with the blood flow to distant structure. For example, in the brain, it can block an artery in the brain, leading to stroke. So that is why hypolipidemic drugs are used to control or to reduce the level of cholesterol, to normalize its level in the blood, including cholestyramine resin, which interrupts the enterohepatic circulation. Cytosterol blocks gastrointestinal absorption of cholesterol. Nevastatin and lovastatin, statin group of drugs, are fungal inhibitors of HMG-CoA reductase. What we call increases MDL catabolism, while nicotinic acid or nicotinamide inhibits glycolysis, thus reduces the flux of free fatty acids in the circulation. This is all. Thank you very much.